Today I'm going to talk about sort of this transfer of metals from geologic to biologic material. So we're going to work from rocks to stalks. I do have a bit of an accent, so I'm originally from Kentucky. You'll probably hear it. One of the best things about that is I can make titles like this rhyme, even though rocks and stalks shouldn't rhyme. But when you say them the way that I do, that sort of lends itself to helping a little bit. I first want to give a little bit of background about some of the stuff that we are able to do in the lab. So even when I was here at Arizona, the analytical side of things always really interests me, and I think the capabilities that we have, both here in Tucson and a lot of other universities, are really, really impressive. And so historically, if we were to think of some of the isotopic systems that we could measure, we were sort of constrained to a handful of what are deemed the traditional stable isotopes or the light stable isotopes. So we've been able to measure things like hydrogen, carbon, nitrogen, oxygen, and sulfur for quite some time. And so we are able to use that information to really understand a lot of different geologic processes, but those do still have limitations. Sometime in the 90s and into the early 2000s, with the advent of new analytical capabilities, we really opened up the entire periodic table, and especially moving towards things that are quite heavy. And so this is the area that I'm interested in. I started getting interested in this area as a graduate student working in the economic geology group here at U of A. Because a lot of ore deposits have very unusual metals. That's why they're interesting. And so I was interested in how we could use some of those more novel or non-traditional isotopic systems to better understand both geologic processes and then ultimately some biologic processes and how those interact. And so what are the applications of those? And so today I'm going to be specifically talking about stable non-traditional isotopes. So these are things that are not radioactive, that are not decaying over time, and so I like to think of them as Skittles. And so you can think of the bag of Skittles as Earth's reservoir of these stable isotopes. And if I were to open this bag of Skittles in this room, we may each have different preferences for the Skittles that we like. I would probably pull out the red Skittles first. Somebody else may pull out the yellow Skittles first because they like them. No judgment, it takes all kinds. But even though we're pulling out unique reservoirs of those colors of Skittles, the overall composition of that bag is not going to change. And so this is how I think of stable isotope systems, is over the course of geologic history, the overall distribution or the ratios or proportions of these stable isotopes are not varying. So we can't use them for things like understanding processes over time. But because of that, there are some sort of regional scale processes that can end up resulting in red or yellow Skittles, but basically the geologic equivalent. And so they can be really, really useful in a range of applications. And non-traditional stable isotopes, now that we're able to very accurately measure them, are incredibly powerful and useful. So many of them are redox sensitive. So they lend themselves to understanding a lot of processes that are inducing different behavior based on different redox conditions. These can be important in ore deposits. They're used very, very often in marine settings, so looking at paleo-redox conditions. Many of these systems are also particle reactive, so that means that they will sort of flock or move to different surfaces depending on what's available, so they can have strong mineralogical controls. A lot of these metals are incredibly integral in Earth surface processes, both geologically but also biologically, so I'll try to convince you of that before we get into some of the nitty-gritty of the talk. Because some of these are metals, they are also vital nutrients for organisms. And so there are biologic processes that can utilize some metals or some isotopes of metals over others. And then depending on the concentration of these metals, they also can potentially pose environmental issues. And so there are lots of different ways that we can view these systems that I think makes them very, very interesting and unique, and is part of why I have sort of flocked to this field over my career. So one of the ways that we can use these is through understanding changes in the environment. And so, for example, it's not uncommon to use these non-traditional metal isotopes to look at the origin of things like ceramic materials, or to try to understand trade routes of ancient peoples. So we can look at things like lead isotopes and ceramics to try to source where the original components of that that pottery or those artifacts came from. That can help us determine if communities were self-sufficient or if they were very trade dependent. These metals can also be great source indicators, so we can understand where metals may be coming from. This is obviously very useful in ore deposits, understanding the evolving chemistry of different depositional environments. And then because many of these metals are also vital components for biologic systems, they're really, really useful in understanding things like metabolism, diet, and mobility. And so these are oftentimes used in anthropology to understand how different trophic levels evolved between different animals and food sources. And so back to this whole spread of the periodic table, it would be really difficult to become an expert in all of these. And so today I only want to talk about two specifically, thallium and lead. And so these, in my experience, go really well together. So they're very near each other on the periodic table. But they serve very, very different purposes or have very different uses, both geologically and biologically. And so what I want to talk about today is how those two particular stable isotopes, and thallium is certainly considered non-traditional, are concentrated or moved or fractionated isotopically, both in geologic material and then ultimately how those geologic controls are translated into biologic material. So I mentioned thallium and lead. I would assume that most of the people in this room are semi-familiar with the lead system, so I will talk about it later, but I won't go into too much detail to convince you of its utility. But thallium is probably an element that you have never thought about, or if you've thought about it at all, it's probably been very rare. And so I want to convince you why thallium is useful. So thallium has some really excellent geologic implications or applications in the geologic realm. The first of which is due to its unique geochemical behavior. So thallium has two oxidation states, a reduced state that is a +1 charge and an oxidized state that is a +3 charge. And it also has two isotopes, a mass of 203 and a mass of 205. And these are really, really heavy masses. So for a very, very long time, it was not expected that thallium isotopes would fractionate or vary whatsoever, and especially not to any sort of measurable extent. So it really wasn't until the early to mid-2000s that groups were even attempting to measure thallium isotopes. So I would consider this still a relatively new field. It's constantly evolving. We're finding new ways to apply thallium isotopes to understanding lots of different geologic and geochemical processes. Because of these two oxidation states, thallium also has two different sizes. So the ionic radius of thallium varies very greatly depending on which oxidation state it's in. And in the reduced state, which is the most common natural state, thallium is nearly identical chemically to potassium. So thallium is obviously not a very common element in geologic materials, but potassium very much is. And thallium can very, very easily substitute for potassium in things like magmatic systems, during hydrothermal alteration, and then also through ore deposition. And so it has a lot of utility there. The chemical similarity to potassium will also come into play once we start talking about the biologic applications here. But it also has some similarities to a lot of other useful and important elements like rubidium, gold, and antimony. Thallium is also incredibly mobile because it is so similar to potassium. It's quite large. It doesn't easily go into a lot of places, so it behaves incompatibly for the most part. And so for a long time, thallium has been used as a pathfinder element for a variety of different ore deposits. So the natural crustal abundance of thallium is quite low, and so it's not too difficult to geochemically find thallium elevated concentrations or sort of geochemical signatures of alteration, and often it's a little easier to find those anomalies of thallium compared to the crustal values than some other systems of metals of interest, especially if they're not going to be near surface. And so thallium is very, very useful in that context. Because of this elemental mobility, it can be redistributed, particularly during fluid flow events, and so that becomes very important, both geologically and ultimately biologically as well. And then we can isotopically fractionate those two isotopes of thallium during a variety of geologic processes. So even though the masses are not very different from one another, depending on the process that's at play, so things like magmatic differentiation, things like hydrothermal alteration, either low temperature or high temperature, it's not uncommon for one of those masses to be slightly preferred. So a little easier to move around, a little easier to fit into a silicate mineral or a fluid phase. And I will talk about fractionation of thallium isotopes. And the terminology is very similar, but different enough from most of the more common isotopic systems that I wanted to put it up here and talk through a little bit. So if you're familiar with isotopic fractionation and how some of those systems are denoted, you typically will see those as delta values. So we're looking at variations in parts per thousand. Thallium varies much less than that. So again, the fact we can measure it at all, I think, is really, really impressive. And so it's showing variations in parts per 10,000, so an order of magnitude smaller. And so rather than being denoted with a delta value, you'll see it denoted throughout this talk as an epsilon value. But really, the gist of this slide is that we are looking at how these isotopes are preferred in one phase over the other. And so we can either have a preference of the latter or the heavier isotope, which would shift things to either positive or negative values. Now, if we were to look at sort of common geologic reservoirs of thallium, there are two that are very, very different from one another and sort of shift the isotopic composition to two extremes. So in the upper right, we have iron and manganese nodules. So these are very, very oxidizing environments. And so the result of that oxidation is a very positive isotopic composition. And then we also have low temperature oceanic crust, and in general, just low temperature alteration. And so these shift things to very negative values. And so this change in redox state is a huge driver of thallium isotopic fractionation, which becomes very, very useful when we think about things like ore deposits. In this diagram, if we were to look at this dashed line, that's going vertical here at about -2, -2.5, that's the bulk silicate earth. And so you can see that a lot of especially high temperature geologic reservoirs don't fractionate thallium very much. Sometimes this may seem disappointing, but in a lot of ways this actually is very useful. This means if we see isotopic shifts of thallium, it's much easier to sort of pinpoint where those are coming from geologically. So a lot of thallium research has been done in marine settings, and they've been able to very successfully track what types of sediment, oceanic sediment, are getting subducted, and even do mass balance calculations to determine how much of those different sediment reservoirs are getting subducted, getting melted, and ultimately coming back to the surface through arc volcanism. Now, on the environmental or the human health side, thallium is also very important. So again, thallium and potassium are nearly identical. So this is them drawn to scale, relative to one another. And so it is almost impossible for biologic systems, plants, animals, humans, to be able to tell the difference between those two elements. Because of that, because so many biologic processes depend on potassium, if thallium is also available, it is impossible to pull up potassium without also pulling up some volume of thallium. And so we will see very, very easy substitution of thallium into biologic systems. And this has some pretty terrible health implications. So thallium is incredibly toxic, so it has toxicity that's akin to things that you're probably much more familiar with, and not for good reason, on the human health side of things, like cadmium, arsenic, lead, and even mercury. And thallium tends to accumulate over time. The body thinks that it is potassium, but it can't use it as potassium, and so it will slowly build. Eventually, this will lead to things like gastrointestinal issues, later nerve damage, and the sort of final telltale sign of thallium poisoning is hair loss. Back in the 60s, thallium was actually used in rat poisoning. It was removed from rat poison because there was a grandmother, I believe in Australia, who got tired of her family. So she used thallium from the rat poison and laced it into her baked goods and was very slowly killing off her family members. And it took several family members before the hospital realized something weird is going on here. Everybody's coming in with nerve pain and no hair. and so she ultimately was caught. But so thallium is very, very dangerous. Now, thallium, luckily, does not occur in very high concentrations in most geologic settings. There are 2 or 3 locations globally where thallium is actively mined because it is a primary component in those ore-forming systems. And those areas are not in the U.S. And ultimately have nearby communities that do suffer from thallium poisoning. Now, because thallium can be easily transferred into biologic material but then not used, thallium also ends up being bioaccumulated. And so what this means is that we can start with geogenic concentrations of thallium that would be of no concern, and that maybe would be even fairly difficult to detect. But plants that are growing in those substrates can continue to take thallium into their systems and ultimately concentrate thallium, oftentimes orders of magnitude higher in plant material than in the soil. And so this poses both environmental concerns, but also great opportunities for things like bioprospecting or phytomining. And so those are sort of the things that I'll get into later on in the talk. And so I think that thallium has a lot of interesting uses, both geologically and biologically. And so a lot of the work that I and my group do is sort of looking at this intersection between those 2 realms. And so that's what I want to talk about today, and then ultimately lead into some of the other metal systems that we're also looking at. And so we sort of look at a few guiding questions to try to understand what's happening here. And the first 2 are the ones I'll focus on initially, which are just understanding how thallium is being remobilized or utilized in geologic systems. So we really want to get a good control on how thallium is moving around, so that way we can better understand if it poses any utility, both geologically, but ultimately if we can track some of those geologic changes into plant material. And so for this was actually part of what I had done as a graduate student here at U of A, was a very detailed mineralogical study through a wide range of ore deposit types to try to understand mineralogical controls on thallium geochemistry, hydrothermal fluid controls on thallium geochemistry, and how those may be utilized geologically. So there was lots of mineral picking, typically by hand, which was a lot of fun, especially when you were trying to pick very light minerals like micas. Very tedious, spent a lot of time under a scope. And then we will work in a trace metal clean lab. So oftentimes we're trying to measure thallium concentrations that are at the part per billion level. And so we work in specialty labs that have essentially no metal components. Fume hoods are plastic, the room is HEPA filtered, light fixtures are plastic, faucets are plastic, eye washes are plastic. And we will take solid rock material, break it down into a solution. We can then either measure that solution directly for concentrations of almost any element on the periodic table, or we can take that solution and run it through column chromatography. And so this allows us to isolate individual elements from a solution so that we have an incredibly pure separate. So in this case, we have these columns. The solution that's yellow is the sample itself. And so as you dissolve material, all of the dissolved ions lead to this sort of yellow coloration. You can maybe see right at the base of these columns, there's a white layer followed by a very orange layer on the top. That is a resin. That's an ion exchange resin. It's essentially little spheres that are chemically formulated to attract cations. And so as we put the sample through, all the dissolved cations stick to those spheres, and then we can pass a series of solutions of different acids or different concentrations to preferentially remove sometimes large groups of elements at a time, sometimes individual elements at a time. And so at the end of this, we will have a solution that should have just thallium left in it. We'll do the same thing for lead. You'll do the same thing for most of these non-traditional stable isotope systems. And so we did this for a whole range of minerals from a variety of different ore deposits. And what we ultimately found was if we looked at coexisting mineral phases, so minerals that were crystallizing more or less in equilibrium during different sorts of processes, that we saw patterns where things like sulfides, which are highlighted here in gray, tend to be much heavier than their coexisting silicates, so things like micas here in red. And so there's a big mineralogical divide, which is dictated by things like the bond type, the bonding environment, how long or short those bonds are. And so there is a very, very notable mineralogical control, and that results in a strong isotopic fractionation across the board, sort of regardless of what our geologic setting is. Now, we also wanted to look at, during hydrothermal events, if there's any sort of correlation between alteration and changes in thallium concentrations or isotopic composition, because some of these processes we'd be interested in both geologically and ultimately biologically could be heavily influenced by fluids. And so here we focused on an area in the Battle Mountain region of Nevada, and we walked through things that were unaltered, which we show up in this sort of black circled region, all the way down to things that are very strongly potassically altered, which we show down in this dark navy region. And so what we end up seeing is that we move from generally low thallium concentrations and more positive isotopic compositions to highly enriched thallium concentrations and much lower thallium compositions isotopically as we pass fluid through. So there's a strong fluid mineral partitioning that indicates that there is a lot of thallium that's being remobilized, and we're preferentially moving one of the isotopes over the other. And so this ends up being useful not only in ore-forming systems, but in things like subduction zone settings, so that we can more easily track inputs and outputs, but also when we look at how fluid is interacting with plants, both locally and regionally. And so if we go back to these sort of guiding geologic questions, some of our takeaways were that thallium does in fact behave incompatibly, so it very strongly wants to mimic potassium if possible. If it is not able to mimic potassium, it will substitute into things like sulfides. It doesn't really like it, and so that dislike sort of leads to this isotopic shift. It can very easily be remobilized during fluid flow events like metasomatism or metamorphism. There is a very, very strong mineralogical control on thallium geochemistry regardless of the underlying geology. And so all of these things sort of lend themselves to now trying to see if that thallium geologic signature, which is pretty consistent across the board, continues to be consistent or translates into biologic material. And so we start to think about things like, how does thallium transfer into plant life, and how is this useful? And so I was really curious in if whether we could use plant signatures, which are much easier for us to sample, much easier for us to digest, and oftentimes can have much higher concentrations of thallium, if those relate to the geology in any sort of predictable way. And so for this, and for even current and ongoing work, we have focused almost exclusively on Brassica juncea, which is an Indian mustard. So Brassica is a family of plants that have shown to be great hyperaccumulators of a wide range of metals. So this means that they can take very low concentrations in the soil and end up with very high concentrations in plant material. Brassica also ends up being full of edible plants, things like mustard, Brussels sprouts, broccoli, collard greens. And so these are very prevalent, both naturally, so the Brassica species is very naturally abundant, but also in people's home gardens. And so this offers an excellent opportunity to try to see if we can have this sort of phytoextraction mechanism through very abundant natural plants and how that may be utilized geologically. So here we started by doing greenhouse studies. Greenhouse studies are excellent because we can essentially control one thing at a time before we get into the natural world, which gets much more complex. And so we were planting Brassica in a soil substrate, except there was no soil. So we were using pure quartz sand that was amended with some fertilizer so the plants could actually survive, and some water-absorbing polymer, so that way they would stay hydrated because there was no organic material to sort of hold onto moisture. And then we can artificially spike or dope that soil or that substrate with any version of metal that we want. So this could be solution-based, This could be mineralogically based, it could be a mixed signature. And so the first set of experiments that I'll show you used both a solution form and a mineralogical form. We allowed these to grow until they reached full maturity, so they got to a point where they were generating seeds for the next generation. Then we harvested and separated those plants into individual plant parts. We then dried those plants in an oven. We ground them down through a series of Wiley mills, so they got into finer and finer particle sizes, and then went through the same sort of process that we did with the geologic material, where we go from a solid plant sample into a solution and then chemically isolated the thallium so we could accurately measure its isotopic composition. And so when we did this, the first thing we noticed is that thallium very drastically increased in concentration in the plants. And so we used 2 different concentrations of solution. So we wanted to start with a solution because we knew the thallium would be immediately bioavailable, which means it's available for plant uptake. One at 20 micrograms per kilogram, or parts per billion, and one at 100. And so if you look at the concentrations in these plants, the actual concentrations are written next to each plant part, and then the color of that plant part also represents the overall magnitude of enrichment. So the darker the shade, the more concentrated the thallium is in that plant part. You will see that some plant parts, especially the leaves, which on this plant are the edible portions of the plant, are very, very, very highly concentrated compared to what our starting concentration was. And so this shows, one, evidence that these plants may be useful in the field for measuring these sort of geochemical anomalies of thallium in a way that we would not necessarily be able to track through just soil sampling, or would be much more difficult to track because the concentrations may be lower. But 2, also poses an opportunity for this to sort of enter the biosphere, which may or may not be a good thing depending on what the goals are. Now, we also did a follow-up study where we looked at, rather than a solution-based thallium, thallium in a mineral form. So does the mineralogical version of thallium, so where it is in a mineral structure, impact how much thallium is available and where it goes? And so here we doped these different soils with thallium in a different mineral. They were all crushed to the same particle size, but other than that, nothing was done to sort of speed up dissolution. We had a feldspar, a mica, and a manganese nodule, and they all were added at the same concentration. So each soil started with 20 micrograms per kilogram, or 20 parts per billion of thallium. And we see very similar trends here that we saw in the solution. So the leaves tend to be the most enriched. For some of these mineral substrates, we see a high concentration factor of thallium in plant parts compared to the soil. But we do see, especially for things like the mica, that there is a sort of dampening of that uptake potential, which I believe to be from things like secondary clays that are forming. So thallium is very particle reactive. If you're breaking down some of these materials into phases that may be able to sort of reabsorb some of that thallium, it essentially becomes much less bioavailable. And so this means that the geology is really critical in the uptake of thallium, and so that may also be able to tell us a little bit about what's happening below the surface. Now, if we were to compare all of these across the board, so here we're just normalizing by taking the overall thallium concentration in the whole plant and breaking it up into where it actually is in that plant, we see very, very similar trends regardless of what form the thallium is in or what our starting concentration is. And so the thallium is mostly moving to things like the leaves, which is quite useful if we want to use this as any sort of field tool, because leaves are things that we can sample and leave the plant intact, which allows us to sort of do either passive sampling or passive monitoring of these systems, passive extraction of metals, and multi-season extraction processes. Now, we took these same plants and we measured their isotopic composition. And so here, our substrate, the isotopic value was 0. So we're starting right at that 0 value. And the darker the color of the plant part, the more positive that plant reservoir is. And again, we see very similar trends. So even though we're starting at 2 different thallium concentrations, we have a very, very positive stem, and then much more progressively light plant parts as the plant continues to grow and develop. And so if we were to look at this in maybe a more easily digestible version, we see a very strong shift isotopically from our starting substrate to the stem, and then everything else sort of starts to shift back into more negative values. So again, very consistent patterns regardless of what we start with, which gives me hope and optimism that we can sort of use these biologic isotopic signatures to essentially back calculate or backtrack what the underlying geologic strata may look like. So in terms of these sort of biologic questions on the thallium spectrum, what we found was that edible plants are able to hyperaccumulate thallium, so concentrate thallium in edible plant parts by sometimes orders of magnitude. That there's a potential environmental threat controlled primarily by underlying geology. So if you were looking at using these plants as sort of a passive remediation tool, which would be very low cost and highly effective, then depending on what your underlying geology looked like, they would be more or less useful. So they may be able to remediate areas very efficiently, or you may have too much of that thallium locked into mineral structures, which also would not be a bad thing because it would require less remediation. This will allow for multi-season sort of phytoremediation or phytoextraction projects, which is what that one shows. And then we can also potentially use these isotopic tools in plants to, over time, track if there are new inputs of these metal sources. So if we can monitor sort of passively over multi-generations or multi-seasons and see these isotopic compositions changing over time, if there are strong shifts one direction or another, that might inform us that there is a new input of these heavy metal sources somewhere nearby, which could be excellent for things like environmental monitoring, but also potential for exploration tools. Now, this sort of led us to start to wonder, thallium is great, but also sort of niche. You know, it's not the most interesting geologically for a lot of people. But are there other heavy metals that may behave similarly, and could they be more useful in things like metal sourcing or phytomining, so trying to find new sources of metals through these biologic means? And so this is where we started to explore lead. So lead is fantastic geologically because lead isotopes are able to really pinpoint unique sources of geologic material and also be able to distinguish between natural and anthropogenic sources. And lead is much more ubiquitous in the natural environment than thallium is, so this makes it a little easier for us to sort of find and measure, and is a great fingerprint of different environments or reservoirs. And so we wanted to then explore if this sort of geologic fingerprinting ability of lead translates into plant material in a way that we could use for things like bioprospecting. And so if we were to think of how this may work, sort of the first thing that I think we need to be able to accomplish through greenhouse studies is to, one, demonstrate that lead is actively remobilized from geogenic material into plant material, and into useful plant material. So if it's being remobilized into parts of plants that are below ground, the roots, for example, which is what a lot of previous work has demonstrated, that's not going to be very useful for us, because we're going to have to extract the entire plant to measure, and at that point we may as well be taking soil surveys. So is it possible to show that lead is actually being remobilized into plant parts that we could harvest individually and leave the plant intact? And then, if we can demonstrate that, can we determine if those geochemical signatures that lead is sort of known for translate in any sort of consistent or predictable way into plant material that we can essentially sort of backtrack? So we did the same thing here. We started with a greenhouse study, but instead of using sort of solution-based lead, we wanted to actually look at different isotopic compositions of lead. And so to do that, we used 3 different NIST standard materials. So the National Institute of Standards and Technology provides materials that they have geochemically quantified. We selected 3 of them that should represent 3 different sources of lead, standard that is representative of mined waste, which would be our natural sort of lead end member. A standard that represents leaded paint, which would be our anthropogenic end member. And then a New Jersey soil, so a soil from near a smelting plant that would represent sort of a mixed end member. Sorry if anybody here is from New Jersey. No knock to them, but this was a potential mixed end member here. And so we planted our plants, same thing, with a pure quartz substrate. We spiked that substrate with these 3 different sources of lead. They're in solid form, they're crushed into a very fine particle size, all added to the same concentration, grew the same brassica plants, and then measured where that lead went. So we spiked these to 50 micrograms per kilogram, so 50 ppb, parts per billion, grew the plants, harvested, and measured. And what we saw, so this is ongoing work, but what we have demonstrated here, not necessarily for the first time, but in a little more robust way, is that we do actually have a lot of translocation of lead from below the surface into above ground plant parts. So there's a lot of lead that moves from the leaves. So you can see through all of these plants, some of the darkest shades of green are in those leaf portions. And so this gave us a lot of hope that there may actually be some really useful utility of looking at lead isotopic signatures in plants, because this would allow for easy harvesting, sort of multi-season studies, and just utilizing these above ground plant parts. And if we do the same thing like we had done before, and we sort of normalize where that lead is going in the different sections of the plant, same thing, the vast majority of this is going into things like the leaves. More often than not, the next largest portion is going into the roots, which is what a lot of other previous work had demonstrated. And so these sorts of plants provide sort of a twofold application for lead. We can extract lead into the above ground plant parts. We can use that to measure geochemically or biogeochemically to understand how lead sources are changing. But we also are stabilizing lead in those root systems, which is great for things like remediation of tailings piles after closure. So let me actually go back here. We're at the stage with this work where we've completed all of the concentration data, and we're maybe halfway through the isotopic measurements of these. So far, it looks quite promising, so I'm very excited about it. But there's not necessarily enough that I wanted to show here. But I'm hopeful that in the next few months, there'll be more here. So if you have questions about it, I'm happy to follow up after this. But from these 2 systems, I think that there is optimism that we can use sort of biogeochemical signatures for things like phytomining extraction or for prospecting. But there are still some questions that I think need to be answered, or some characteristics that need to be better quantified. One of which is how things like other changes in geochemistry, spatial or temporal variations may impact these biogeochemical signatures. So far, everything we've done in the greenhouse has been sort of in an individual plant basis, but in the natural world, you have no idea in some cases how long that plant has been there, or how far away from a source is. So how far can we get, or how many generations in can we go and still have those biologic signatures reflect some of that natural geochemistry? So we have ongoing trial work and greenhouse studies that's looking at these sort of spatial and temporal variations. We also need to understand how greenhouse results may compare with field-based studies. So obviously the field gets a lot more complicated. You can't control for all your variables. And so we have ongoing work in the field, some of which is in Montana. IU has a field station outside of Bozeman that happens to be near several legacy mines. They're small, they're sort of artisanal mines, but there's now a lot of naturally developed plant life there, especially in the brassica family, and so we've been measuring samples there to see how accurately we may be able to pick up signatures of where those original tailings are compared to upstream or downstream and what that looks like. And so there is sort of a longitudinal study translating some of this work from the greenhouse into the field. And so with this, I do think there's a lot of hope here, a lot of optimism that this may be a really interesting application to sort of blend the geologic and the biologic sides in a way that is cost effective, is fairly low energy, and can be sort of a passive system that you can look at how it evolves over time. I could not end this without thanking the lab members who have done a big bulk of this work. So these are a couple of master's students and an undergrad who have sort of spearheaded some of the analytical work and the greenhouse work. I did some greenhouse work here in Arizona. I'd argue that it's much worse working in a greenhouse in July here than a greenhouse in July in Indiana, but they still put in a lot of time and effort and energy. And so I'm really, really grateful for their time and effort. And with that, I would be happy to take any questions. Yes. Oh, sorry, can you ask it into the microphone so the folks online can hear?
Hi. I was wondering, the plants you were working with, were they generally annuals or perennials? And how do you sort all that out?
Yeah, that's an excellent question. So these are perennials. They will come back year over year. I think that obviously we're focused on one specific plant. I talked about how sort of prevalent brassica is in the natural environment. I specifically chose this particular brassica species because it is one of the few species that is sort of naturally abundant in the arid Southwest, which is where a lot of my interest lies. It's very hard for some plants to grow and thrive here, but it seems to do a pretty good job naturally. But I think that there is a lot to be said for expanding into other plant types that are also as abundant or even more abundant, depending on the geographic region that you're interested in working in.
So did you have an explanation to why the leaves get concentrated more than other parts of the plant?
Yes, that's also an excellent question. So because, at least in the case of thallium, it mimics potassium so closely that it is moved very similarly as potassium. And so potassium tends to be moved with a lot of other nutrients through uptake of water and nutrients through the roots, through things like the leaves, where then that water evaporates off. When that water evaporates off, or transpires off, typically the potassium that's left behind then gets utilized through things like energy production. That's not going to be the case for thallium, and so when that water leaves, that thallium is just left behind in those leaves where it concentrates. It seems like lead is doing the same thing, even though it's not going to be mimicking potassium, but it's moving through that same sort of solution system that's getting a lot of materials into the leaves. I think that there's actually a lot of ongoing work, not in my group just yet, that's looking at these same sorts of questions and applications, but for things like critical metals. So rare earth elements are a big area of interest right now for a variety of reasons. And so things like brassica have not really been demonstrated, at least things like mustard plants, have not been demonstrated to be really great at taking up a lot of rare earth elements and moving those into the leaf portions. And so a lot of that research has had to focus on other plant parts or plant materials. And so I think that depending on the metals that you're interested in trying to capture, one, they may be in various plant parts, and two, they may need other types of plants or other species of plants to really be able to be extracted.